Hello, and welcome to the Rejuvenation Roundup podcast, where we explore the latest in life extension and anti-aging science with a dive into a month's worth of insights and new breakthroughs. This podcast is a combined effort of the Lifespan Extension Advocacy Foundation, which operates Lifespan.io, and Future Grind, a podcast that explores the ethics and implications of emerging science and technology. I'm Ryan O'Shea, and I'll be your host. Let's take a look back at the updates from September. Kicking things off with our research roundup, a recently published review explains why fecal microbial transplantation, or FMT, might be able to help sufferers of Alzheimer's disease. The gut and brain are closely linked through biochemistry, but this review highlights research showing that gut dysbiosis may be a key player in Alzheimer's disease. The researchers outline a chain of biological relationships. Gut dysbiosis increases the level of harmful compounds in the gut, decreases beneficial compounds such as short-chain fatty acids, and negatively impacts tight junction proteins, which impact intestinal permeability and directly affect the blood-brain barrier. Increased intestinal permeability has a negative impact on inflammation. Bacteria crossing the intestinal barrier have been shown to lead to the release of inflammatory cytokines. These cytokines lead to immune cells going to where they are not wanted, in addition to microglia becoming triggered by inflammation, breaking down the extracellular matrix and causing brain cells to suffer dysfunction. This triggers known pathologies of Alzheimer's disease. With this in mind, several research groups singled out what they believe to be the bacterial culprits in this chain of events. Directly replacing the identified bacteria in the gut is an approach worth exploring. 2 different mouse studies have shown that fecal transplantation can improve spatial learning and memory, as well as performance on the well-known Morris-Maze test. One transgenic mouse study showed that FMT can decrease inflammatory cytokines, partially restoring the microglia in the brain. Another compelling study also involved transgenic mice, showing that FMT decreased both tau and 2 different amyloid beta proteins while improving cognitive abilities and neuroplasticity, reflecting the brain's ability to learn new things. While the reviewers do not cite any study showing that a dietary change can ameliorate Alzheimer's in humans, they do cite a previous review showing that omega-3 fatty acids, antioxidants, a reduced intake of animal proteins, and reduced fat and sugar are linked to a decreased incidence of Alzheimer's disease. This shows the possibility of prevention rather than a cure. Another team found that long-term caloric restriction can decrease the bacterial culprits involved in the cascade. Researchers have developed a biological aging clock based on movement data from wearable devices. Using their clock in the same dataset, they identified a few possibly life-prolonging nutrients and drugs. The researchers trained their model, which they called MoveAge, on data from 2,634 people over the age of 18 using a machine learning method, and then they tested it on a different part of the dataset. The model proved to be fairly predictive of chronological age and general mortality in people aged 40 to 70. A strong negative correlation was observed between biological age and the consumption of fiber, magnesium, and vitamin E. Correlation was weaker with drugs, but one candidate did emerge, doxazosin, which is used to treat benign prostate hyperplasia and hypertension. Finally, the researchers tested the effect of doxazosin on C. elegans worms, an animal model widely used in geroscience. The scientists were able to achieve a modest lifespan extension in worms treated with the drug. Unfortunately, MoveAge was trained on data from just 1 week of physical activity. Still, its success hints at the potential of such clocks. MoveAge offers us a glimpse into a future in which multiple types of data will be routinely gathered and cross-referenced by billions of devices worldwide. This will deliver a clearer picture of our health and help advance medicine, including personalized medicine, using the power of machine learning. According to new research, it may be possible for humans to live 130 years, or perhaps longer, even without a lifespan intervention, but the chances of reaching that age are slim. The new study, published in the Royal Society Open Science Journal, examined new data on people who are 105 or more years old. Based on their findings, the risk of death ceases to rise beyond a certain point, and plateaus at around 50-50. This means that beyond the age of 110, the risk of someone dying with each passing year is essentially a coin toss. The researchers suggest that if there was a limit to human lifespan below 130 years, they should have been able to spot it using the new datasets, but there was no such indication that this was so. However, even if it is theoretically possible that humans can reach age 130, the odds are very much against it. The study authors note that without major medical advances, the human race is unlikely to go beyond 130. This increases the importance of longevity science. And now for some news nuggets. On the heels of their COVID-19 vaccine approval, Biotech mRNA cancer treatment has entered human trials. Underdog Pharmaceuticals has concluded a $10 million financing round, and a new cryptocurrency, Transhuman Coin, has made its debut. As a research and charity-oriented token, a portion of every transaction is allocated to fund science and technology initiatives. 4 more videos from our recent Ending Age-Related Diseases conference have been released, including an interview with Terry Grossman, a talk by Steve Horvath on pan-mammalian epigenetic clocks, a talk by Aubrey de Grey on emerging challenges to longevity science, and one by Alexandra Stolzing of the SENS Research Foundation discussing what SENS is doing in terms of grants, company creation, and intervention development. Sticking with video, new episodes of Lifespan News have been released, including a recap of Aubrey de Grey's talk at our conference, some insight into the roughly $26 million in impetus grants recently made available to longevity researchers, and a video on the brand new Jeff Bezos-backed longevity company Altos Labs. Here's Brent Nally to tell you a bit more about that.
There's a new company focused on solving aging. Investors are reported to include billionaires Yuri Milner and Jeff Bezos. In recent months, there's been some hints, but considerable secrecy surrounding the formation of a new aging research company by some prominent members of the biorejuvenation community. Well, it appears the mystery of this new large-scale venture has finally been solved. The new company is reportedly called Altos Labs, apparently named after Los Altos, California in Silicon Valley, where Russian-born Yuri purchased a mansion in 2011 that made national headlines due to the $100 million sale price, which was the most expensive single-family home ever purchased in the United States at that time. It's important to understand that Altos Labs hasn't made an official announcement yet, so we're doing our best here at Lifespan News to get you this video based on current available information, and we encourage you to check our sources in the video description as always. An article recently released in MIT Technology Review by Antonio Regalado revealed much of the details of Altos Labs that we're sharing in this video. Yuri is one of 2 confirmed billionaire investors of Altos Labs. Yuri was an early investor in Facebook and has a net worth currently estimated at about $5 billion according to Forbes. The other billionaire investor is reportedly Jeff, the world's current wealthiest person with a net worth of about $200 billion according to Forbes. Jeff founded Amazon in 1994 as CEO and remained CEO until Jeff stepped down to become executive chairman on July 5, 2021. A securities disclosure filed in California in June 2021 indicates Altos Labs has raised at least $270 million. Incorporated in Delaware and in the UK earlier in 2021, Altos Labs plans to establish several institutes in places including the San Francisco Bay Area, San Diego, Cambridge, UK, and Japan, and is recruiting a large group of university biorejuvenation scientists with lavish annual salaries around $1 million, plus potentially Altos Labs stock and the promise that they can pursue unfettered blue sky research on how cells age and how to reverse that process. Longtime longevity enthusiasts will no doubt be familiar with past formations of seemingly similar companies such as Google-backed Calico, which appeared with a great amount of hype about how Google was taking on death about 8 years ago in September 2013. I remember I was super excited when the Calico news broke. While considerable fanfare heralded Calico's arrival, very little in terms of tangible rejuvenation tech has resulted, unfortunately. Instead, Calico has produced fundamental research about the biology of aging, which of course has a valid place, but such research does not exactly live up to the expectations that the longevity community had for Calico in late 2013, especially considering the enormous resources Calico has spent compared to other much less well-funded entities focused on solving aging. That said, the one exception to this was Calico's recently published work about safer cellular reprogramming, which may prove useful in the future. If you'd like to learn more about this, check the video description to read Lifespan.io's article by Arkady Mazin. So how might Altos Labs be more successful compared to Calico? Well, unlike the fairly vague goals that accompanied Calico's arrival, Altos Labs has set its sights on a promising target, cellular reprogramming, which has been shown to reverse epigenetic alterations in mice, which we've reported here at Lifespan News. Click the card above or check the video description to watch that episode. Some great researchers are reported to be involved in Altos Labs. Partial cellular reprogramming pioneer Carlos Espuza Belmonte, a Spanish biologist at the Salk Institute, is rumored to be joining Altos Labs. Belmonte famously demonstrated that living animals could have their cells rejuvenated using partial cellular reprogramming in 2016. While Salk has not confirmed this yet to the best of our knowledge, if Belmonte is on the Altos Labs team, that gives us more confidence in this venture leading to something useful. Steve Horvath, pioneer of DNA methylation clocks, the gold standard for aging biomarkers, is also reportedly joining Altos Labs. Steve has a wealth of experience regarding aging biomarkers and has spent years constantly improving and refining aging clocks to support the progress of our field. Shinya Yamanaka, who shared a 2012 Nobel Prize for the discovery of cellular reprogramming, will reportedly be an unpaid senior scientist and will chair Altos Labs' Scientific Advisory Board. It's very likely this roster of researchers will continue to grow in the next few months, so make sure you're subscribed with the bell turned to all notifications for updates. While it's tempting to judge Altos Labs on the current disappointment of Calico, personally, I'm excited about Altos Labs. I can't wait to see how this story develops because I believe Altos Labs has the right focus and some of the right people. And it certainly has raised a significant amount of money thus far, with a realistic path to raise much more money considering the world's wealthiest person is invested. Another non-governmental entity that has massive funding and can potentially receive even more massive funding is the Chan Zuckerberg Initiative, or CZI, which was founded in 2015 by Facebook founder and CEO Mark Zuckerberg and his wife and medical doctor Priscilla Chan, who are also co-owners and co-CEOs of CZI. The power couple reportedly plan to make an investment in CZI of 99% of their wealth from their Facebook shares over their lifetime, thus setting up CZI to likely be one of the most well-funded philanthropies in human history, and setting an example of philanthrocapitalism. The CZI's mission is, quote, to build a more inclusive, just, and healthy future for everyone, end quote. CZI states, quote, we support the science and technology that will make it possible to cure, prevent, or manage all diseases by the end of the 21st century. We foster collaboration between scientists and engineers, develop new technologies, and build support for basic scientific research, end quote. Here at Lifespan News, we've also reported on the potential for Elon Musk's Neuralink to help improve healthy longevity, and Elon said this in 2020.
With an effort that's not too crazy, you could probably stop aging, reverse it if you want.
We'll be keeping a close eye on Altos Labs, Calico, CZI, and Neuralink to see how things develop. I'm very excited to see some of the world's wealthiest people creating companies focused on solving aging, which is responsible for about 110,000 daily deaths globally on average, out of about 150,000 total daily deaths globally on average.
Science to Save the World has also released 3 new episodes on topics such as tiny medical robots that could one day invade our bodies, hydraulically controlled robotic hearts, and cellular reprogramming as a method of potentially reversing biological age. Here's the episode on cellular reprogramming, which discusses the importance of Yamanaka factors.
Could reprogramming old cells make them young again, leading to potential cures for age-related diseases? In 2006, a study by doctors Kazutoshi Takahashi and Shinya Yamanaka showed that it was possible to reprogram cells using just 4 master genes: OCT4, SOX2, KLF4, and CMYC, or OSKM for short. These 4 reprogramming factors are often called Yamanaka factors. A somatic cell is any cell of a living organism other than a reproductive cell. Before the Yamanaka discovery, scientists assumed that egg cells, or oocytes, contained a complex array of factors needed to reprogram a somatic cell into becoming an embryonic cell. After all, they thought, the feat of reprogramming an aged egg cell to make a new animal must be controlled by many factors present in the egg cell. Takahashi and Yamanaka turned this idea upside down when they showed that just 4 Yamanaka factors were needed to achieve this transformation. They used these factors to reprogram adult mouse connective tissue cells, or fibroblasts, to an embryonic state called pluripotency, a state where the cells behave like an embryonic stem cell and can become any other cell type in the body. In 2011, a team of French researchers, including Jean-Marc Lemaitre, first reported cellular rejuvenation using the Yamanaka factors. During their life, cells express different patterns of genes, and those patterns are unique to each phase in a cell's life from young to old. This gene expression profile makes it easy to identify an old or young cell. At the time, it was also known that aged cells such as fibroblasts have short telomeres and dysfunctional mitochondria, 2 of the 9 proposed reasons we age. Jean-Marc Lemaitre and his colleagues tested the effects of Yamanaka factors on aged fibroblasts from normal old people and also from healthy people over 100 years old. They added 2 additional pluripotency genetic factors to the OSKM mix, namely NANOG and LIN28. They discovered that together, the 6 factors were able to reset cells from old donors back into a pluripotent state, meaning that they could become any other type of cell in the body. These became known as induced pluripotent stem cells, or iPSCs. The researchers noted that the cells had a higher growth rate than the aged cells from which they had been reprogrammed. They also had longer telomeres, as well as mitochondria that behaved in a youthful manner and were no longer dysfunctional. In other words, reprogramming the cells reversed some of the aspects of aging and rolled the cells back to a state similar to that of development. The final step for the researchers was to guide these iPSCs to become fibroblasts again using other reprogramming factors. The result? The reprogrammed fibroblasts no longer expressed the gene patterns associated with aged cells, and had a gene expression profile indistinguishable from those of young fibroblasts. Essentially, they showed that epigenetic alterations, or changes to gene expression patterns, a reason we age, were reversed. In addition to this, they showed that telomere length, mitochondrial function, and oxidative stress levels had all reset to those typically observed in young fibroblasts. Telomere attrition and mitochondrial dysfunction are 2 more reasons that we are thought to age. This was the first evidence that aged cells, even from very old individuals, could be rejuvenated. This was followed by a flood of independent studies confirming these findings in fibroblasts and other types of cells. So, could Yamanaka factors be used in living animals? Stay tuned for our next episode to find out.
That's it for this episode of the Rejuvenation Roundup podcast. Thank you very much for spending another month with us and for your help in the fight against age-related diseases. Whether you're donating, spreading the word, or simply listening to our content, we appreciate your help. Remember to subscribe, leave a review, and post about us on social media. This will increase our reach and introduce more people to the importance of life extension science. Don't forget, you can get additional deep dives into science, technology, and futurism on the Future Grind podcast. Find out more at futuregrind.org. Once again, I'm your host, Ryan O'Shea, and on behalf of the team at Leaf, we wanted to thank you for joining us. We hope to see you next time on the Rejuvenation Roundup podcast.